Okay, proper technique for streaking a blood plate with bacteria.
When streaking plates, you always want to make sure that you're labeling the very back of the plate, just in case your lid comes off or gets misplaced or anything, and you know what organisms you're working with. We're going to do this just like we did the yeast. We're going to sterilize our loop, let it cool for a little bit, grab the right organism. You don't need much inoculum. We're going to do our first quadrant. First quadrant should always come down to about halfway. I want to make sure to streak between. Normally, if I were doing this, I would try to keep the plate covered a little bit between each streak, but since we have to teach you guys, do 3 streaks across for your second quadrant. Sterilize your loop. Wait a little bit. You can always test the loop against the side. You can see it seamed up a little bit. Drag it across 3 more times for your third. And then make your fourth quadrant.
Okay, so we'll be going on now to unknown number 3. What we're going to be doing is looking at a group of bacterial organisms that'll be a mixture of gram-positives, gram-negatives, and some that will have different characteristics that you'll have to identify using certain types of media and using the gram stain. Your first step that you'll be doing in this lab will be to streak a blood plate for isolation, which we covered in the previous video. The other medium that you'll be introduced to in this lab will be McConkey agar. This is a selective and differential agar that will give you a certain color reaction if your organism is a lactose fermenter. First of all, only gram-negative bacteria will grow on McConkey. The gram-positives will not grow. If you get a pink colony, a bright pink colony produced, that means it's a lactose fermenter, and this medium is used typically to detect E. coli in samples. So when you streak the McConkey plate, you'll do it exactly the same way you did the blood plate. You're going to heat your loop in the bactocinerator. You're going to pick up a culture from the tube that we've given you. We'll have 5 different organisms here. And then you'll streak one quadrant, you'll flame, streak another quadrant, heat or flame, and then streak the 3rd and 4th quadrants on your McConkey agar. Now, the organisms that we're going to be looking at are Bacillus, Staphylococcus, E. coli, Salmonella, and Pseudomonas. So you'll be given 5 different tubes with the pure cultures of those organisms. The point of this whole exercise is to just familiarize you with using a flow chart, which you'll find on page 12 in your laboratory guide. So we're going to look initially at growth on McConkey agar. If it does grow, then you go to your positive. Yes. Does it ferment lactose? In other words, does it produce the pink colony? If it does, then it's an E. coli. If it does not, you come down to the next part of the chart where we'll inoculate a TSI slant, and you'll read that reaction. I'll go over that in just a second. The other part, you're looking at no growth on McConkey, and then you can assume it's going to be a gram-positive. If you get no growth, then you're going to look at that with the gram stain to see if it's a gram-positive rod or a gram-positive coccus. So all those illustrations will be in your Moodle site, where you can look at the still shots of these different reactions. Now, the TSI slant is a very useful medium for differentiating organisms, gives you a lot of different kinds of test results. It's inoculated by using a straight needle. You'll go into your unknown culture, or in this case, we know what we're dealing with, but you will be getting unknowns. You'll go in, you'll take a sample from that slant with the needle, and then you're going to inoculate the TSI by stabbing it straight down through the center, almost to the bottom of the tube, but don't touch the bottom of the tube, and then straight back out, and then you're going to also streak the slant. So you'll be streaking the slant and stabbing down through the center of the medium. And you'll be getting different color reactions on that media that tell you a lot about which sugars the organism utilizes, and if it produces gas, and if it produces hydrogen sulfide. All those photos are in your Moodle site that you can look at.
Okay. As always, whenever we go to streak a plate, we're going to want to write on the back or the bottom of the plate the organism, and if you were in class, your initials, so you don't get it confused with somebody else's. Every time we go into stock agar, we must sterilize our loop, or we pick something that's pre-wrapped and sterile. Let it cool a little bit. And go into, grab a little bit. I'm going to streak this just as we would the blood agar for isolation. And that's it.
This is the inoculation of the TSI slant.
This is once more we go in. Go down, not all the way to the bottom. Once we're out of the agar, we streak the top. That's it.
So the TSI is going to be incubated at 37 degrees for 24 hours, and then we'll look at the reactions.
So we're going to go over gram staining. In order to prepare your slide, you want a very small amount of water on the slide. From your blood agar plate, you would select a small colony and mix it in the water. Now you want this kind of cloudy. You want to be able to see that there's bacteria there essentially, but you don't want it completely opaque. If it's too opaque, then you might be just overrun and not really be able to tell the size and shape of your bacterium. That I would usually let air dry for a little bit, and then once it's air dried, which it's starting to get, if you can tell in the center it's still a little wet, but once it's completely air dry, you want to bring it over on top of the Bacti-Cinerator and just kind of go over the hot portion to heat fix it. It's important to heat fix it because if you don't, then your bacteria is just going to slide right off your slide when you try to do gram stain. So I got my slide here. We're going to flood the slide with crystal violet for 30 to 60 seconds. For the sake of the video though, we're going to go a little bit faster than that. Once the 30 to 60 seconds is up, we're going to rinse the slide with cold water. Then we're going to flood it with iodine for a minute. And gently wash with cold water. Now we're going to do this for 10 seconds or until it runs clear, essentially. But you don't want to over decolorize your slide. And then we're going to apply our counterstain saffron for 30 seconds. Once the 30 seconds is up, then we're going to rinse again with cold water. After that, we blot dry with paper. And that's it. Okay. So we go through our crystal violet step for 30 to 60 seconds. We wash with cold water. Then we flood the slide again with iodine for 30 to 60 seconds. We wash again gently with cold water. Then we proceed with the acetone for about 10 seconds. And then we flood the slide once more with saffron for 30 seconds. So once your slide is dry, you're going to want to drop one little drop of immersion oil onto it so you can actually visualize under the scope. From there, you're going to want to see what the predominant color is going to be. If it's gram positive, it'll be purple. If it's gram negative, it'll be pink. It'll hold that saffron. And then you're also going to want to look at the shape and the size, relative size of your organism. Is it rod? If it's short rod, sometimes that can be confusing to look at because it looks like cocci, it looks like short circles. But just when you're looking at it, you have to be aware of this is a three-dimensional object. Even though it's tiny, it's three-dimensional. And so you really need to look at overall how all the organisms look.